Okay, so now I'd like to introduce Keith Mansfield. He's the Director of Discovery and Investigative Pathology, an awesome title, at Novartis. And before that, he spent very many years at Harvard and as a pathology faculty. Today, he's going to talk about multimodal considerations for understanding the molecular basis of morphological changes in animal models for precision disease modeling. So welcome.
Thanks very much, Melissa. Yes, I'm a veterinary pathologist. I'm currently at Novartis Institutes for Biomedical Research in Cambridge, Massachusetts. So I'm going to talk about some of the molecular pathology approaches we can use to evaluate precision models, animal models. Sorry.
Uh-oh.
The green one advances and the red one goes back.
Okay, great. Thanks very much. So molecular pathology is focused on the study and diagnosis of disease through the examination of molecules in tissue sections or body fluids. And it attempts to understand the molecular basis of morphological or structural changes in tissues and ties those then to functional alterations. And it requires a firm foundation in anatomic pathology. By nature, it's multidisciplinary. It integrates data that comes from a variety of sources, and it uses a number of tools, including immunohistochemistry and in situ hybridization, along with advanced imaging modalities. Molecular pathology is frequently used in the diagnosis and management of patients clinically, and it's a discipline that's one of the foundations of precision medicine. Molecular localization is the ability, again, to define the spatial distribution of molecular entities like proteins or nucleic acids in tissue sections using tools like immunohistochemistry and in situ hybridization. And this allows the pathologist to begin to understand how alterations in the spatial orientation of these molecules ties to functional deficits in the animal. Immunohistochemistry has advanced quite a bit over the past 10 or 15 years. There are now a number of these automated staining platforms that are available that have really improved efficiency and reproducibility of the assay. When I was a postdoc, we did this by hand. It would take 10 to 12 hours hands-on to complete an assay. With the automated staining platforms, that time is reduced to about 1 or 2 hours for a particular assay. And the machines can run 24/7. So it turns what was a very labor-intensive assay into something that can be used in a high-throughput format. I think a shortcoming of these assays still is the quality of commercial antibodies, and these can vary widely. And an enormous amount of work has to be placed in validating an assay before it's actually used to assess tissues in an experiment or in a patient. And this sort of acid validation would include things like Western blot, flow cytometry, staining of cell lines with known expression values, comparison to mRNA expression, peptide blocking experiments. In the end, there's no single gold standard to assess the validity of an immunohistochemistry assay, and one has to integrate from a number of different sources. In our experience, for novel targets in particular, most commercially available antibodies are not specific or sensitive for the target. In situ hybridization is the other tool that we use fairly routinely, and again, this has been adapted to the automated staining platforms, and one can use traditional sorts of probes like riboprobes or DNA probes, but within the last 10 years or so, there's a new generation of probe technology that has been advanced by Affymetrix and advanced cell diagnostics that have enhanced sensitivity and specificity. And essentially, these platforms use a series of oligopairs that align on the target nucleic acid linker, and then a amplification chemistry that produces a discrete dot at the site of hybridization. And thus the assay has resolution down to single molecules of RNA within the tissue section. And one can see here in the middle panel an example of a target expressed in hepatocytes in association with non-alcoholic steatohepatitis, and adjacent to that, a double label in which we can see the orientation of the mRNA expression adjacent to areas of fibrosis. The technologies also allow one to look at splice variants. So on the panel on the right-hand side, one can see a differential splice variant expression of a target, one variant expressed highly within enterocytes of the small intestine, and the other variant expressed within the myocardium. So again, these new technologies can be applied in an automated format and can be used in a more high throughput sequence. Another technology that's underdeveloped that's been very useful is that of highly multiplexed immunohistochemistry assays. So the traditional assays generally will allow you to multiplex 5 or 6 different antibodies together, but in these newer platforms termed image mass cytometry or laser ablation inductively coupled plasma MS imaging, which actually is a nice descriptive term of it, one can multiplex 20 to 30 or even more markers together. And this utilizes rare metals to conjugate to the antibodies. The pool of antibodies can be incubated in with the tissues. And then 2D spatial orientation is maintained, and one can get a virtual reconstruction of the orientation of those labels. This sort of technology requires significant resources to validate the protocols, and bioinformatics are critical in extracting the fullest amount of data. So it's not, at this stage, ready for routine use, but when one is interested in looking particularly at immune cell subsets where a number of markers must be used to identify the cells, for example, Treg cells in tissues, these particular platforms have a lot of utility. I think a resource that is sometimes forgotten but critical in this idea of comparative molecular pathogenesis are biobanks that contain well-curated collections of normal and diseased human biological samples, including FFPE or formalin-fixed paraffin embedded samples, and then also samples from preclinical species and precision models that we'll be using. The comparison of the human disease samples to samples from precision models can determine the similarities and differences in the morphology and molecular alterations, and this comparison can inform the selection of the models that most closely parallels those aspects of the disease that one wants to investigate. This assists in experimental design and also in the end can reduce the total number of animals needed to complete the experiment. So by using archived animal model tissue plus the diseased human tissue, one can assess whether the model is going to actually adequately address the questions that one is interested in. And this is an example of such comparisons. The NADP oxidases contribute to TGF-beta induced fibrogenesis, and in several disease entities we believe that this pathway is operating, particularly in pulmonary fibrosis and in certain renal diseases. We had a question whether this pathway might be operating in hepatic cirrhosis and whether this pathway may allow us to intervene in human disease. And so we could compare both baseline expression of different NADP oxidases by in situ hybridization. And what we see in both mouse and human is that in baseline expression is rather limited. We find it identified particularly in the Kupffer cells of both species. In a series of animal models that we looked at that were associated with bridging hepatic cirrhosis, we can see that there's a marked upregulation of P22-FOX and NOX2 in these hepatic septa, but this is not recapitulated in the human sample, suggesting that the mouse models would not adequately predict response to therapy that one would expect to see in humans. As another example, we can look at the morphology of solid organ cancers, and in this case, look at pancreatic adenocarcinoma, which is actually morphologically a complex lesion. We can think of it as being composed of both epithelial and non-epithelial components. The epithelial component will include the neoplastic cells themselves, and in addition, the residual acinar and ductular cells that are undergoing degeneration because of the process. There is also an immune component that's quite complex. It can be comprised of both immunoreactive, or cells that would be beneficial to the progression of the tumor, as well as immunosuppressive cells. And then finally, we have a stromal component that would include, again, neoplastic elements that have been derived from epithelial mesenchymal transdifferentiation, as well as non-neoplastic components such as tumor-associated fibroblasts, endothelial cells, lymphatic endothelial cells, pericytes, and adipocytes. And so we can ask, how well do mouse models reproduce these elements of the microenvironment? In this slide, we can show a comparison between human pancreatic adenocarcinoma and a couple of xenograph cell lines that are commonly used in NSG mice to assess response to therapy. And by looking at H&E and markers for epithelium and stroma, we can appreciate relatively rapidly that there are marked differences in the morphology. These kind of well-formed and mature septa that form within pancreatic adenocarcinoma of the human samples are largely absent. We have abundant EMT, epithelial-mesenchymal transdifferentiation, in the animal models, but again, the mature fibroblastic septa are lacking. Additional markers, we can see that there's an absence of T and B cells in the NSG mice. While macrophages are present, they don't show an activated phenotype, and the overall proliferative rate within the neoplasms are substantially higher than what we see in the human clinical samples. A new precision model that's being developed is the use of organoids, syngeneic organoids, in which genetic engineering has been used to introduce changes in known oncogenes and tumor suppressor genes, and these models actually reproduce the morphology to a greater extent. We get the development of the mature fibrovascular stroma. In the case of immunocompetent mice, we get the infiltration of T and B cells with the formation of tertiary lymphoid structures. Macrophage activation is similar, and the overall proliferative rate is more similar to what we see in pancreatic adenocarcinoma. And this is not to say that the xenograft models don't have their utility, but we need to understand how differences in morphology may impact the relevance of the disease in modeling human cancer. Another advance in molecular pathology is that of digital pathology. And in a traditional workflow, tissues are collected, they are processed through paraffin and then stained, and really a process that's not changed in over 100 years. The slides are delivered to the pathologist then reads them through a traditional microscope, the slides get archived, the pathologist writes a report. And it can be very difficult to then gain access to those archived slides again. Much of the information is contained within that slide is not actually reflected in the report, and being able to access, again, that information or that data is difficult. In a digital approach, instead of delivering the slides to the pathologist, slides are scanned, and then these scanned images reside in a database along with the associated metadata. And this has several advantages for the pathologist. They can review the slides at their desk, they can take pictures relatively easily. But the big advantage is that these slides can be accessed again relatively easily. The associated data, the metadata is contained with these slides, and one can apply additional bioinformatic approaches to interrogate the slides. So the pathologist can look for a particular morphologic diagnosis, recall all of the slides that had this diagnosis, and look for associations with patient metadata. The real advance will occur as we apply deep learning to these slides to generate novel patterns that can then be associated with diseases. The digital approach also facilitates whole slide image analysis, and this allows us to quantitatively measure staining intensity, cell number, cell morphology, the spatial relationship between cells. In this approach, the slides are stained by immunohistochemistry on an automated staining platform. They are scanned. The software can easily be trained to distinguish the neoplastic versus non-neoplastic elements, and then to segment the cells, look at their staining intensity, and generate H-scores or other measures that can be compared quantitatively. Tissue microarrays also fit within this workflow. So tissue microarrays can be used for both immunohistochemistry and in situ hybridization on these platforms. The TMAs can contain 24 to 100 or more cores. They are arrayed on an individual slide. And you can create custom arrays that are focused on a single species, on a single organ, a single disease process. The quality control of the source material is paramount, and we find it was often lacking in commercially available TMAs. And so internal creation of the arrays is beneficial because it allows the pathologist and the histotechnologist to work closely together to provide an array that is most useful. And the example on the bottom right-hand side, we have a single species array that's been stayed for alpha-smooth muscle actin and Ki67, and then spectrally resolved to show the relative orientation of those two markers. Image analysis can also then be applied to the tissue microarrays. So as I said, when you have these automated platforms up and running and you are assessing tissue microarrays, you can assess literally thousands of tissue cores in a single day. Those cores still have to be assessed, and the image analysis can help us with this. So the slides, again, are scanned. Image analysis softwares can identify the cores and assign those cores and perform cell segmentation and staining intensity. This can then generate H-scores, which can be heat mapped and allow one then to correlate the individual scores with different aspects of patient or subject metadata. Image analysis can also help with the spatial resolution of target expression, here showing an example of mRNA. Again, the newer in situ hybridization techniques have resolution down to single molecules of mRNA. And with image analysis, one can develop mRNA counts per individual cell. This can be kind of difficult to interpret in a low resolution picture of the tissue, but using image analysis, we can then actually heat map the level of mRNA expression per cell and observe differences in a macro scale. And in this example, we can see downregulation of a particular target and control in treated animal rather easily. A newer technology that's being developed is termed spatial transcriptomics, where we can actually profile several thousand genes within a single slide and resolve the spatial orientation of changes in the gene expression. Genomic profiling can also be integrated with molecular localization, and this can assist in elucidating the pathogenesis of disease. We often take paired samples, one for genomic profiling, one for routine histopathology and molecular localization. And the analysis of these samples can be complex because the overall gene expression changes may result from alterations in individual cells, or probably more importantly from changes in the cellular composition of the tissue. And molecular localization studies can then be used to confirm expression changes in the cellular source. And in the example on the right, we have accumulation of foamy macrophages within the subscapular sinus of a lymph node. Gene expression studies indicated several targets that were differentially expressed in the lymph node. We could then go back and localize those targets to show that targets were expressed in the abnormal cell type. Genomic profiling can also be used for cell type related signatures that can be extracted from complex tissues. So in this example, we have a kidney and we're looking at the expression of nephron, a podocyte structural gene. And we can see that in normal rodent kidney, there's some variation in the expression of this particular marker. And this is due to the relative proportion of glomeruli within the sample. One can use hierarchical clustering techniques to then identify additional genes that correlate with nephron 1 across these gene types. And one can see that there are kind of three major classes of genes that are identified in this approach. And these then genes can be mapped to particular morphologic structures, to podocytes, mesangial cells, and glomerular endothelial cells. So again, novel cell type related gene signatures can be extracted because of this variation. And then we can then go back and localize these particular genes and confirm that the cell type related signature is as we expected based on the clustering. So in summary, molecular pathology advances our understanding of the comparative pathogenesis of human disease in relevant animal models. It's a rapidly advancing field that kind of integrates traditional anatomic pathology skills with molecular biology and bioinformatic approaches. And the interrogation of tissues from these models, from precision models, really should be made in conjunction with the evaluation of human disease tissues to better understand the model's relevance and limitations. And molecular pathology is at that interface and well suited to do that. Future advances will build on the use of bioinformatics, particularly highly multiplexed localization assays, and also computational interrogation of digital slide databases. And I'll just quickly acknowledge some of my colleagues who have helped with some of the work they've shown today.